introducing Cari to you on the occasion of the defense of her master's thesis. Cari comes to us from Coastal Carolina University, which is in Conway, South Carolina, and she did a marine sciences bachelor's degree there, which involved many things, but among them was a project doing some scuba field work on urchins in Jamaica, and that got her interested in the sea urchin system. She's also done diving at many other places, including Catalina on this coast. And so she decided to come out west, and since coming to Arcata, Cari's had many exciting and important life changes. I think I'm not talking just about harassing sea urchins there. Okay. Basically, though, I want to point out that she's chosen a project that looks at an aspect of urchin biology that was essentially unknown 5 years ago. And so this has meant some troubleshooting, some trial and error, some changing plans, and basically Cari's positive attitude has been great throughout all of that. So without further ado, take it away, Cari.
Thank you, Ed. Ed, would you mind popping the top light switch for me? Thank you. Great. Well, thank you all for being here today. This is very exciting. I get to share my master's work with you. I'll start by introducing two species of sea urchins that live right on our rocky reefs. We have the red sea urchin, Mesocentrotus franciscanus, and the purple sea urchin, Strongylocentrotus perforatus. These two urchins live together on our reefs. Many of you have seen them in the intertidal. Typically, you find red urchins in the subtidal, in the lower regions, and the purple urchins are more intertidal. Red urchins can actually outcompete purple urchins for space, for advantageous kelp, for lower wave action. And this is interesting because it kind of puts them in their two own little zones. Red urchins have longer spines and larger body sizes, which is actually why I chose to use them in most of my research. It's interesting to note that red urchins recently were reclassified into Mesocentrotus genus. Historically, they were always placed with Strongylocentrotus, but some phylogenetics research has put them into their own genus, Mesocentrotus. So today I will introduce the topic of dermal vision in sea urchins. I will then go into chapter 1 of my thesis, which was its own project, and then chapter 2 of my thesis, which was its own project, spurring off of part 1. And then I will introduce some suggestions for future research if someone might want to pick this up where I left off. So let's start by talking about the external features of a sea urchin. When you strip away the spines that protrude out from all angles of the sea urchin, you are left with a test. And that's actually a calcareous endoskeleton here. And there are two important regions on the sea urchin. We have the interambulacral region right here. You can see these plates that fit together well that have these little nodules, and that's where the main spines come out of the urchin test. And then in between those regions, we have the ambulacral region, and that's where actually all of the tube feet are. Tube feet are very important for locomotion. They're very important for sensory. They actually reach out in front of them with their tube feet. You can see this guy in the background actually was reaching out his tube feet into space to feel around. And the tube feet are most importantly how they stick to their surface. So they're actually little suction cups that they adhere to the surface. And tube feet are only found in these 5 ambulacral regions. The pentaradial symmetry is really exhibited with the tube feet and interambulacral regions. So these little spiny spike balls running around the seafloor are, for all intents and purposes, pretty primitive. They don't have eyes. They don't even have a brain. However, they do have the ability to detect light. There are photoreceptors in their skin all over their body, within their spines, within their skin on top of the dermis there, and in their tube feet. So this is exciting. It means their entire body, this dome shape, this hemisphere, is a photoreceptive unit all in itself. So we've known for a long time that they will tend to move towards shade when sunlight shines on them in the intertidal, but we never knew they had exciting visual abilities until 2010. And it turns out that sea urchins can see. In 2010, a group out of Duke University found that when they put a tropical species of urchin in the middle of a tank and shine a light from above, they can actually detect a dark spot placed on the wall. So sea urchins, with a very strong correlation toward either toward or away, moved toward or away this dark spot. So it meant that they can actually detect that dark spot that's approximately the size of their own body. That's fascinating. This means that they have spatial visual capabilities. I put this little biologist down here because in this study, the biologist was actually underneath the tank, looking up through a transparent bottom to observe the animal's movement. So it's pretty funny. Another aspect about this study was that it was actually a round tank, which is great because it allowed the urchin freedom to move in any direction. But the animals chose to move directly toward or move directly away from that black spot. And in this case, the researchers posited that perhaps it was because they were interested in the dark spot or motivated by fear in some way to move away. We should also note that they used white light from above to illuminate the space. There was no wavelength variability. It was just a full spectrum white light. So we've established that there are no eyes, no actual eyes on the urchin, no ocelli, not even a primitive eye like a sea star. But when the purple urchin's genome was sequenced in 2006, the scientists found something interesting. They found opsin. And opsin is a very important gene in visual capability. It's actually the reason that we detect light and the reason that we detect color. We don't understand why they have this capability. Why is this in their genome? So it was a mysterious finding, and we're not entirely sure what the adaptive significance is. But from that, it has spurred a few other intriguing questions that we'll talk more about. We should also take the opportunity to remind you that as an entire photoreceptive unit, this animal can kind of function like a hemispheric retina. Okay, so our eyes are powered by retinas, and this hemispheric shape actually allows you to take information from all angles around and on top of this hemisphere. Some other examples of hemispheric retinas, hemispheric shapes, are found pretty exclusively within the animal kingdom, but the most advanced eye is on the mantis shrimp, and humans have actually based Blu-ray technology off of the mantis shrimp eye. We also see a couple other interesting hemispherical arrays. This is actually a solar panel that has been created so that you never have to tilt it toward the sun. Instead, it absorbs sun from all angles throughout the day. Pretty brilliant, really. We also see a brand new camera that actually has, each of these little domes is its own camera, and that's designed around a fly eye, the ability to take in information from 360 degrees around the hemisphere. We have a robot designed to interact with people who are struggling to communicate with other people. And then another example finally is, of course, a security camera. So you can see all around Target what's going on at all times. Another example that I just came upon of a hemispheric eye being so useful is actually a vacuum that has never been released. It's coming out soon from Dyson. So if you need the ability to leave your house and come back with a clean floor, you can check out Dyson's new vacuum. The most fascinating thing about it, though, this is the whole vacuum. The most fascinating thing is this little convex hemispheric camera on top that will actually memorize your living room based on what it can see around you. So a picture frame on the wall, a cabinet against the wall, you'll be able to see the camera can see where it is and not bump into anything. So that's pretty exciting. And finally, we should of course point out the hemispheric shapes on top of robots here from Star Wars. BB-8 is a good example, but if they really knew what they were doing, they would have put hemispheric cameras all around his eyeball, all around his head. So we should keep in mind that it's interesting that these animals have the ability to detect light all over their body. Now, the green urchin is a pretty ubiquitous urchin all throughout the world, and they were studied for their ultraviolet capabilities to detect ultraviolet wavelengths. And this particular urchin has the very cute behavior of covering their bodies with detritus from around themselves. I've actually even seen green urchins with the bodies of their dead friends on top to cover themselves. And in this study, the scientists used various wavelengths of UVA, ultraviolet A, which is closest to our visible spectrum, UVB, and then they also used visible photosynthetic light. And when photosynthetic light was combined with UVA and UVB, they saw the most significant covering behaviors, meaning that instead of just being triggered by photosynthetic visible light, ultraviolet inspired them to cover their bodies. Ultraviolet can be very damaging, as we know from multiple sunburns, and ultraviolet may be damaging to invertebrates living beneath the water surface. So we talked a little bit about opsin, how it's the key protein in color vision. A study out of Caltech in 2011 came about just as I was starting my research, actually, and found opsin throughout the animal's body. So not just photoreceptors, but opsin genes. And they found the strongest, the highest correlation, the highest density of opsin in the tube feet of the animal. So this is the end of a tube foot. It's actually the suction cup that adheres to surfaces. And you can see the end of them right here. These little suction cups are at the end of the tube feet. And that's where the highest concentration of opsin is, which is fascinating because, again, why do they have it and why is it being expressed? So in that same study, the scientists put the animal in a long runway tank. They placed the animal, the purple urchin, into the center of the tank, and then they shined lights out of one end, and the urchin had the freedom to move toward or away, but they couldn't move side to side. So in this case, the animal is about halfway, and they were allowed to move toward or away from whatever wavelength of light was shining here. Now they tested several wavelengths. They exposed the urchin to red, orange, green, and blue light, and then an all-spectrum white light, as well as a dark control where the animal was placed in as usual, but no light was shined. So their results are on the graph coming next, and we see some interesting patterns. Let me orient you to this slide here. We have the baseline where the animal would have been placed right here at 0, and the positive numbers are the centimeters that it might have moved toward the light up here. Different wavelengths are listed here, or away from the light. For example, if the animal were placed here in the center during a red trial, each of these bars is an example of one animal and its movements. So about half of the animals moved toward red light and half moved away. Same is true of orange and green trials. However, during blue trials, the purple urchins moved away from the blue light, which is fascinating. Only one animal moved at all toward blue light. So this shows an interesting pattern. Again, why are they responding to just blue? What is it about blue light that motivates them to move away? So this particular data set is interesting. The authors don't really address why this data set has such movements moving toward a light that's off. But as we'll see in the future of this study, it's a little bit mysterious about what animals do in the dark. So this brings me to my first set of research questions. I wanted to point out that red urchins are at the far end of our visible spectrum, and purple urchins are at the other far end of our visible spectrum. So it's possible that perhaps their abilities lie within their body color. Perhaps there is some correlation to what they detect and what their body color is. So I wanted to know if red and purple urchins respond oppositely to the same color stimuli. We know that purple urchins respond to blue wavelengths. Do red urchins respond to red wavelengths? And finally, I also wanted to know, do red urchins respond at all? Maybe this is just a purple urchin thing. Maybe only purple urchins have the ability to detect different wavelengths. So I designed a study that would allow me to put each animal in a test tank and test its abilities. So each red urchin received 3 minutes in a light-proof chamber. I used 1 urchin per trial, and I measured two variables. I measured the distance moved and then their angle of movement relative to the light. Each animal was was kept in their own cubicle in case I needed to reference what animal was behaving in what way. For the experimental light treatments, I used LEDs. LEDs are inexpensive. They come in many sizes and shapes, and their wavelength bands are very narrow, meaning that there's not a lot of other colors that we might be incorporating into our experiments. So I used red light, blue light, ultraviolet light, and a control treatment where the light apparatus was placed into the tank, but the light was not turned on. This is an example of my test chamber. This was a plastic chamber, 1 meter in diameter. You can see the radius here is 50 centimeters. And at the beginning of the trial, the urchin was placed here in the center. And for example, at the end of a trial, an animal might be toward the edge. This is just one example. Now, the experimental light was random. The location of the experimental light was randomized throughout the trial, but I always measured the angle of the animal's final movement to where the light was. And the light was actually kind of in the form of a lightsaber. There was a clear plastic tube with 6 LEDs mounted in the bottom so that it would shine directly out at the animal. Wherever the lightsaber was placed, it was oriented toward the center of the tank. So the animal could move freely within the tank, but I opened the lid and turned off the lights after 3 minutes. And then I measured where the animal landed. That's actually what these 2 little weights are. Is the meter stick on top of the tank had 2 little movable weights, and that allowed me to measure both the center of the tank and the angle from where the animal moved. So this is an example of all of my results from red, blue, and control trials. I'd like to take this opportunity to point out that it's pretty confusing. So this is actually a graph that I wrote the code for in R with the help of Mark Rizzardi. And at that time, there was not an R package to display polar data, circular data. So we actually wrote this code back in 2013 to display my data visually, because I can have a collection of numbers, that's great, I can have a collection of vector data. However, I don't know what it really looks like. So we wrote this code simply to display it. And for example, the animal would be placed in the center here. If this is the test chamber, the animal would have been placed here. And each line represents one animal's movements in a coordinate direction. All of the data are adjusted as if the light were pointed from up here and the animal were placed here. You can see that we have a lot of variability between the different color treatments. So I promise I cleaned it up for you. This does pose a problem though. The statistics that you might normally use on a linear data set don't work here. You have a totally different set of parameters for circular data. So I had to scrounge and find some other circular statistics tests. And with the help of Katie McDonald, I went into circular statistics. Yes?
I just wanted to ask, did they just start moving right away? I know there's no clump in the middle suggesting that some animals are just sitting there. Do they automatically just start booking in one direction or another as soon as you put them down?
In this study, most of them did. There are a couple of vectors that are just, they stop right about here or right about here. But this is a 3-minute trial. So as soon as 3 minutes were up, the trial was over. And if they didn't move very far, then that's what we got. This is a measure of 50 centimeters. So quite a few of them didn't move very far.
And could they have gotten much further in 3 minutes than the perimeter? Or was that about as fast as they could go?
That's about as fast as they could go. This urchin right here actually started to climb up the wall, which is why you see it going slightly past 50 centimeters. I measured it at 51.
Okay.
Yeah. So I needed to find a branch of statistics that would suit circular data. I split up my ultraviolet vectors from the rest of the data, and you can see here that within the 4 quadrants we have representative vectors. So the animals moved into all 4 quadrants during ultraviolet trials, which is interesting. And this is just an example data set, not real data, of what a clustered data set might look like. What you're looking for in circular data are clusters, so vectors that are clumped together. And the test for that is called a Rayleigh test. It actually tests for clustered data. And so in this data set here, we see a Rayleigh with a p-value equal to 0.84. Very random. Very little, no significance whatsoever. And then on a data set like this, where you have all of the data spread nearly between just one quadrant with maybe a little bit of scatter, you get a Rayleigh that's highly significant with a p-value of 0.001. So this shows that there's very little chance that these were random movements. We also have a second test, and the V-test is actually what tests for whether that cluster is truly around the mean of the data. Perhaps that cluster is a part of a larger data set, and it's not really the mean average of that data set. So the V-test ensures that your cluster is around the mean. Okay. So my hypothesis for chapter 1, when red urchins were exposed to red, blue, or ultraviolet lights, I predicted that their movements would be unequally distributed, and that we'd see that with the Rayleigh test. So my results for chapter 1 are displayed here. Again, let me orient you. So in each graph, we say that the light source, whether it was on or off, is up here at 0 degrees. Each line represents one urchin's movement throughout a trial. And then we have their distances moved in centimeters along the x and y axis here. And of course, these are the degrees that the angle measurement that they would have moved. So in the control trials, we see a pretty nice scatter of 10 urchins, movements into each quadrant, so we would not expect a clustered data set. In red trials, we again saw movements into each quadrant with a small, interesting little cluster here. Not sure if that's significant. Ultraviolet, we already saw the p-value for that, if you recognize this graph. We know it's highly insignificant. And then with blue trials, we saw animals moving mostly south of this border. So if the light's up here, the animals mostly moved away from blue light. And again, this is red urchin data. So when I ran my statistics, I saw no significance in control trials, which was a relief, because that meant that I didn't have any sort of environmental or tank bias going on within my testing chamber. Red light trials were also not significant, so I did not see any significant clustering in the animals' movements. Ultraviolet light, definitely not clustered, which was a little bit surprising given the results from the green urchin, the tropical green urchin. I was thinking perhaps we would see some interesting movements, some clustered away from blue light. I'm sorry, from ultraviolet light. And then we did see a significant cluster when we used the Rayleigh test with a p-value of just barely 0.049. This was 10 urchins though, so if I had increased my sample size, I expect that the p-value would have been more significant. It was enough prompting though to move forward with blue light as an interesting tool to make urchins move, to make them move in a consistent pattern. So this was the first record of wavelength detection in red urchins. That's exciting. It was also interesting to note that red and purple urchins behave similarly in blue light. So instead of behaving oppositely at opposite ends of the spectrum because of their body color, instead they actually behave similarly. So that was unexpected, but again, very interesting. And again, it was surprising that the animals, the red urchins, did not behave consistently in ultraviolet light. Perhaps the ultraviolet light that I used was not quite the right wavelength. Perhaps they would behave if it were a more damaging wavelength into the UVB range, or even UVC, which is the most damaging kind of ultraviolet light. But it does, again, give me some evidence to move forward, a lot of evidence actually, to move forward in testing red urchins with blue light. So, take a water break real quick. In part 2 of my study, I wanted to use blue light as a probe, just like an optometrist might use a light in your eye to make you blink or to make you respond. I wanted to use blue light as a way to test animals' behavior, test their capabilities. This idea of dermal vision is a pretty new field with urchins, but there were some very early studies in the 50s, in the late 50s, looking at the ability to detect light with a tropical urchin, the diadema species. It's actually the species that I worked on in undergraduate. So this diadema species was used to determine what parts of their body might have been the most susceptible to light. And in this study, the urchins were actually cut up, and the scientists removed portions of their body while the pieces were still moving, because urchins do that, kind of like zombies. And so their spines would still move in response to different wavelengths. So they shined a variety of different wavelengths on the aboral interambulacral region, which is the top side of the animal, in between where the tube feet are. So they measured spine waving responses with different wavelengths. They shined red and orange and green and blue wavelengths. And they found, actually, that when they shined blue light on this cut portion of the test, they found a stronger response with blue light. However, it was a cut up piece of an animal. It wasn't a live animal. It certainly wasn't necessarily behaving like it would in the natural world. So a little bit hard to swallow that idea. But 2 years later, these authors were back at it with diadema, and they cut out more pieces of the animal. But this time they made a map of their findings. So they used both portions, both the interambulacral region and the ambulacral region, where the 2 feet would be, or where these 2 red lines are. And the map that they made shows that the dark areas are where they saw the most spine waving, and these dotted areas are presumably where they saw the least amount of spine waving. And there's a pretty small sample size here. Again, it's a cut piece of animal, and we don't really know what wavelengths they used in this study. It was an all-spectrum white light. So it's something to question. It's something to wonder, okay, if they think they saw a stronger response in the ambulacral region, perhaps that is the tube feet responding because of the opsin that has more recently been found. So this gave me a little bit of a starting point where I wanted to improve on both my study from part 1 and their studies, and try to find a way to test a live whole healthy urchin for its abilities in blue light. So I'll talk to you a little bit about my purposes here in chapter 2. In chapter 1, I was able to illuminate the entire half of an animal because, for example, say the light source were up here, 6 LEDs were shining, and it illuminated the entire half of the animal, which is great. We saw a response from that. But I also wanted to be able to map red urchin responses by using small point source pieces of light on different parts of the body. I wanted to be able to elicit reactions from different portions of the animal's body. For example, perhaps I put 2 little lights right here on the ambulacral region to really stimulate that area. Might I see a different response if I put 2 little lights in the interambulacral region, or perhaps along the top here? There are many different places that I could put lights on an animal, but I wanted to try and parse that out. So if I use blue light as a probe, I can perhaps map this urchin's response. Now, this is actually a picture of one trial, 10 seconds apart, where I put 2 lights on an animal. So this is just a quick intro to what I did in the future. I also wanted to be able to ameliorate some of my handling effects. Throughout sea urchin history, animals have been scraped out of their tank with a spatula. Who's done that? Yeah, you got a spatula and you scrape it up, and you typically leave some 2 feet behind, which is just another way of severing an animal's body. So I wanted to try to avoid at all costs severing the animal from parts of its body. So I designed a little plate system where these large pieces of black plastic were clipped to the side of each cubicle, and each animal was kept independently again. When I wanted to remove an animal, I simply unclipped the top and removed the whole plate so that the animal was placed into the experimental tank on its substrate. No 2 feet lost, theoretically. So that was a big improvement. I also wanted to be able to track urchin movements throughout the entire study. In my first chapter, I put the animal in, closed the lid, and didn't watch for 3 minutes. So I really have no idea what an animal was doing during those 3 minutes. Perhaps it zigzagged and had a little dance party. I don't actually know. So I wanted to be able to see the animal's movements throughout the entire trial. And this meant either being present or having a camera, and I chose both. So I was able to watch the animal's movements throughout the entire light trial. This is a great picture of my setup. I spent a lot of time in a dark tent. Most of you probably saw it up at the Marine Lab. The dark tarps up on the scaffolding up there. I had a tank that I was able to fill with the same amount of seawater for each trial, and no white light entered the chamber. But I did have these 2 safe lights, these 2 darkroom photography lights. And the light that came out of those was just enough to illuminate for photography and allow me to see what I was doing. But the wavelength of that light was between the red and orange that had been tested both by me and by Ulrich Luder. And neither of those wavelengths inspired any kind of response in purple or red urchins. So I was able to use that in good conscience, knowing that it shouldn't deflect the animal's movements. So I had 20 red urchins up at the lab, and each animal received each light treatment. There was total darkness except for the Kodak safe lights shining down, and you can actually see these 2 spotlights, basically, that again allowed for photography. I had my GoPro camera mounted over top, this is one example of an actual photo that I used in my data. And I chose this photo because it's evidence of my Taylor Swift dance parties during trials. And then you can also see the blue light attached to the animal's body here. The wires coming out attached to the power supply. So I'll explain exactly how I attached these lights. Each light assembly here weighed 1.5 grams, and it consisted of an LED light wired to the power assembly. Thank you, Lewis. He wired these lights so that they were waterproof, It's a small LED, like you saw earlier, a very small LED, that was actually covered with a sleeve to limit its diameter. The diameter of this light was only 5 millimeters. And then this black plastic electrical tape allowed me to stick it to this piece of floral foam. Now the floral foam is a little bit silly, but it was the only substance that would stick to an animal's spine throughout an entire trial. And so it's the kind of foam that florists use to arrange flowers, and it's just sticky enough to stick onto the scales of an urchin's spine. And so these pieces were one use only, so I used several hundred of them throughout my study. But that meant that I was able to push them onto the spine of the animal and direct the blue light exactly where I wanted it to go in each trial. This is a great time to point out that red urchins are much bigger than purple urchins. You're able to really see this ambulacral region here, ambulacral tube feet. It might take a little bit of practice, but you're able to find the tube pretty much in the dark on a red urchin that it might be harder to do on a smaller urchin. So once these lights were pushed onto the animal, I would place it in the center of the tank, turn on the camera, turn on the lights, ready to go. So I was left then with a series of photographs. And these photographs were my data. They were essentially living vectors of my animal's movements. And this is that same picture we saw earlier, overlaid with some of my analysis. So each photo was uploaded into my imaging processing software. And this is actually 2 photos taken 10 seconds apart. Okay? And the way that I was able to measure their movements and measure the angles was where I took the center point of where the light was shining on the animal, and I drew a line that was its predicted path if it were moving 180 degrees away from blue light. So this red line here is where I predicted it would go if it were moving 180 degrees away. This is slope 1. Okay? I had a series of equations that allowed me to detect the angle, or to measure the angle of where it actually went. So this photo is 10 seconds later, and the center of that blue light is point B. And so this angle here is what I measured, the difference between where it should have gone and where it actually went. This allowed me to have a predicted direction and where the animal truly went. This means that trigonometry came in handy. We always wondered when we might use that. Came back to bite me. And Sokotoa was my best friend. And then I, of course, incorporated the Rayleigh's test as well as my circular means and V tests to process this circular angular data. Thanks for sticking with me. Okay, so this is a great example of an actual urchin trial. So again, these photos are 10 seconds apart throughout an entire trial. And the animal is placed here on its little plate so that it's happy and intact. And then every 10 seconds, a photo is taken. And this is an example of the idea that animals don't move in a straight line during a trial. It's not a straight line. If the animal were moving exactly away from blue light at all times, it would have started here and moved approximately this way. But in each interval, in each instantaneous vector, I'm able to pile these on top of each other to get a large data set for each trial, which is fascinating. You're also able to see when I ended a trial, which is when the animal hit the side of the wall with either a tube foot or a spine, the trial came to an end. So, how do I map the behavior of these animals? I have my photographs as data. What do I do with them? So I have all of my angles in my Excel spreadsheet, and my animals tracked along the tank, and these are 2 examples of the directions they might have taken. They might have gone in a very straight line, or they might have gone a little bit back and forth, kind of like my last urchin. And so this is just an example of what I thought might happen. I thought if I illuminated the entire half of an animal with very bright, very concentrated light, perhaps I'll get a very tight cluster. Just an example. Or if I give it less stimulus in a smaller range, perhaps perhaps I'll have a wider spray here of still clustered, probably still significant, but maybe less so because it gets less information. So this is how I thought I might be able to use this data. My hypothesis for part 2. If I used 2 blue lights to illuminate an area in the ambulacral region, I predicted that their movements would be unequally distributed again, and that, of course, my Rayleigh test would show that. So, take a breath. These two graphs are an example of, are actually real data that show where the animal moved in the tank. There's no light information at this point. We're not talking about where the animal moved in relation to light. Instead, we're talking about where it moved relative to geographic north. And this is simply to eliminate tank bias again. So I created these vectors, or these plots with my R code to show that the final destination of each animal were really spread throughout the tank. Now, I need to point out that each one of these lines is actually a composite vector. So each of those little squiggles that we saw is every 10 seconds, right? And so then I would average that entire group of vectors to make one final mean vector for a whole trial. Okay, so my mean vectors are plotted here throughout the tanks, and I was happy to see no significant tank bias whatsoever. The animals all moved throughout the tank quite equally. And then I looked at my data for my actual experimental results. And when I shined the blue light on this end of the Urchin's body, the animals moved pretty much away. That's a pretty great cluster right there. We see that that's really away from blue light, right? That's pretty exciting. My p-value is very significant. My v-test is very significant at 3 degrees. My mean vector is 3 degrees. So that's great. However, my control trials, where the light was not on, the lights were on the animal, but the lights were not turned on, was also pretty significant. So that was surprising, obviously. But at the same time, we can still use this. This is interesting information. The Rayleigh test was just as significant with a mean vector. I'm sorry, the mean vector was at 340, which is just about right here in this space. So not entirely sure what caused the leftward shift there, and we can talk more about that later. But surprising results out of animals in the dark, once again. So I decided to try and troubleshoot this. Maybe I could parse out the effects of whatever was bothering the animal to move so distinctly away from the light assemblies. So I tried an opposite side treatment, where I put 2 light assemblies on in the ambulacral region, but opposite each other. Perhaps blue light could motivate the animal more than a light that was off, than an assembly that wasn't on. So if blue light were truly more motivating, the animal would move approximately in this direction. Okay? If these 2 light assemblies were equally annoying, they might move in this direction and split the distance. Okay? So I wanted to try and tease out these 2 effects. So this is a tank bias test again. I went ahead and plotted the vectors, the mean vectors, with the example that 1 light was not on, 1 light was on. And this shows, again, that there was no bias within the tank motivating the animal. But in this case, they did go ahead and split the distance. So this shows that the blue light and the no light treatment caused them to split the distance and move away from, as far away from those 2 stimuli as possible. So I ran the statistics again, of course. The average vector was 5 degrees away, off of center here. So to summarize my results so far, my animals were successfully photographed in very dim light. They were tracked throughout the entire trial, and my analysis worked really well. All of my analysis methods were great. You can actually see the remnants here of how I was measuring angles in my image processing software. You can see the safe lights, and you can see the habitat plate right there that was left behind. There are definitely some possible reasons for this unexpected behavior, where the animals were just as annoyed by blue light as they were by the light assembly on its own. It's possible that what I used, the components within the light assemblies were toxic or were annoying to the animal. I used this kind of floral foam because it worked really well. And then later I found out, after all of this happened, I found out that there's actually formaldehyde in floral foam. So that's a potential motivator. Research has shown that invertebrates will move away from, they'll move out of the area of caffeine or formaldehyde. So that was something that I could have checked out earlier. We also had 3 different kinds of adhesive on the burritos that I made, named by Katie back there. These burritos had a piece of masking tape to allow me to tape the electrical tape onto the little piece of floral foam. So there are certainly some chemosensory possibilities for the behavioral bias here. And I'm not entirely sure what caused the animal to move to the left in my control trials. If you think that if it were just bothered by the light assembly, just as bothered by the light assembly as they were blue light, they would have behaved identically. However, I know that it wasn't any kind of environmental bias, like a running water that was next to my tank, or anything outside the tank, because the tank controls were all nice and random. So if I were going to continue doing this, and I were going to continue to try to map an animal's response to blue light, I came up with some new techniques. I would try to eliminate external stimuli entirely. I wouldn't put lights on the animal again. I would continue to try to make sure that they stayed intact, use my tile method, my little plate method of removing the animal. I might even try to test them in their habitat. Perhaps it would be best to keep them, if space permitted, in their individual test tanks and start a trial right where they are all the time. I would continue to use my analytical methods that I developed, but I would test a new light delivery method. So I decided that the best way to do that would of course be with lasers. So I would get a focusable blue light laser, I would mount it overhead of the tank next to the camera, and it would be on one of those nice gooseneck arms that's really stable so that I could point it at the animal and point it in the area of the animal that I'd like to test. And because it's a focusable laser, I would be able to change the diameter of the beam. So I could try larger diameters, smaller diameters. I could even get 2 lasers, and I could put them on different parts of the animal. I could do a lot, or one of you could do a lot with lasers and blue light. So I wanted to review my conclusions with you from my thesis. From part 1, I learned that red urchins will move away from blue light. I learned that red urchins, surprisingly, did not move away from ultraviolet light. That is something that I would also pursue further. I would try a lot of different ultraviolet wavelengths to try to get them to move. In part 2, I learned that urchins don't travel in a straight line. In fact, a lot of my urchins kind of went back and forth and around sometimes during trials. It was very interesting to watch. I also learned, I developed a great technique to continuously track urchins' behavior throughout a trial. I wanted to say thank you to very, very many people. First, my very patient and supportive advisor, Ed Metz, to my great committee, Bruce, Ethan, and Brian. I'm looking forward to all of your comments. Mark Rizzardi really helped me with R, as so did Allison Brokaw, and they basically taught me how to use R. Katie McDonald suggested circular statistics, and I couldn't have done this without circular statistics. Tim Bean and Adam Cummings actually fixed my final horrifying problems right at the end with statistics. My brother Nick and Christine Garcia really taught me how to use Photoshop, which I appreciated. Grant and Dave up at the Marine Lab are, of course, fantastic. They built me everything I might need to house an urchin. Liz and Cheryl, and now Stephanie, do everything for us, and I really appreciate them. Lewis and Marty wired my LEDs and printed little 3D plastic pieces to contain my bulbs. And I also received funding from the HSU Biology Master's Grant, twice. And I also, of course, appreciate all of you for being here today. And my husband, James, thanks for all of the back rows. And thanks to you for being here. What questions do you have, Shanna?
I thought it was interesting at the end when you were talking about lasers and moving forward with being able to pinpoint a laser. My question is, do they spin when they move? So if you have a laser that's pointing on this one spot, not only will they be moving away from it, in a straight line, but will they do they turn their bodies often or frequently?
That is a great question, and that was actually one of our main concerns. Pardon me while I scroll through everything. That was one of our main concerns as we developed this project, because we know that as a radial animal, it can do whatever it wants. And so we tried to be able to do it this way so that we could track all of the possibilities. With a laser on a gooseneck, I could change it. So as it rotates, the idea is that it would be stable when I set it, but very movable throughout a trial. So I could keep continuing to move it. With a red urchin, you can see exactly where you want it to be, so I could track it, just like I did here, but without touching it.
Okay.
Thank you. Good question. Yes, Steven.
Have you thought of any reasons for adaptation for moving away from blue light, like gorging, or maybe I know they spawn blue, but I don't know much about sea critters. But I don't know. Have you thought of any reasons why they might just move away from blue light?
So many. But no good ones, really. The best theory is simply that opsin is a very complex protein, and it's a very complex piece of a genome. To only have an opsin that would code for one wavelength would be advantageous, because you only have to keep track of one. But it also would allow you, if you pick the right wavelength, it would allow you to track all light. Because in the ocean, blue light penetrates the furthest. All other wavelengths are absorbed. So if you can only track one, if you can only detect one wavelength, the best one to be able to detect would be blue light. Maybe? Yep. But real quick, what was also interesting about the Ulrich-Luder study, the first study that looked at wavelength, I don't know if I can get out of this. Let me find that real quick. It's much better illustrated by them. In this graph, I found it interesting that they found no response to green and orange, because urchins' primary food is kelp, and it's kind of a greenish-orange color. So they weren't responding to the wavelengths that might point them towards good kelp.
That's kind of what I was,
Yeah, so that's interesting too.
Would you think that would be more like a chemoreceptor response rather than
It could be, absolutely, because they have great chemosensory abilities. But if they were using their color detecting abilities to find food, you would think they would respond, right? So they must not be using their visual capabilities to find food. Angela?
With your burritos you added up, was there any like spine shedding or anything like that of like behavior of them trying to remove them?
They could occasionally pop off one of the burritos. One of the great things about red urchins is that they have the longer spines, so it usually took longer than the trial would last for them to pop them off. Sometimes they would. By spine shedding, do you mean like would they lose a spine? No, they never lost a spine to try and get one of these off of their body. They would occasionally be able to push them off with other spines though, using multiple spines to shed the. Yeah, there's a lot of trial data I didn't use.